Hello, hi, good afternoon, Shubha Lakshmi. Can you please confirm whether I'm audible and visible? Shubha Lakshmi, are you there? Yes. Shubha Lakshmi, can you hear me? Okay, means am I visible also properly? Okay, we'll wait for some time for others to join, then we'll start. Some brief introduction about me. Good afternoon to all, and good afternoon to all. Welcome back to Good afternoon, all. Good afternoon. Ladies and gentlemen, let's begin with a short introduction about me, myself. Hello. Good afternoon to all. Let's begin with a short introduction about me. Myself, Kajal Kachawa. I'm PMR Research Scholar from IIT BHU Varanasi. I will be your TA for this particular course, that is host pathogen interaction, for the duration from January to April 2025. So I will be taking classes on every Saturday from evening 3 to 5 p.m. And this is the week 4 class. And the previous 3 week classes is already released on the YouTube. So if you have not watched that, you can check it out. Till now, the week 5 content is released on the NPTEL portal. So you can visit the portal and go through the whole content. For week 1, Sir has covered history and development of immunology. For week 2, immune organs are covered. In week 3, cells of immune system is covered. And for week 4, the cytokines and other immune mediators topic is covered. And recently, the week 5 content is released. And in this week 5 content, the innate immunity and the pattern recognition receptors are covered. So these 5 week content is already released, and the assignment for week 5 is also recently released, so you can check it out. In last class, we have taken the assignments for week 1 and week 2, and in today's session, we will be taking the week 3 assignment. So this is your complete course. In the previous class, we have discussed some topic relevant to your assignment problems, where we have seen the life cycle of lymphatic filariasis. In this case, what happens is the this mosquito there, this elephant, this particular disease, that is the lymphatic filariasis, is spread from the, this is also known as elephantiasis, and this particular disease is spread from this particular worm, that is filarial worm. The name is Wucheria bancrofti and Brugueria malai or Brugueria timori. And this is a vector-mediated disease which is spread by the by the bite of a mosquito. These particular mosquitoes can be Anopheles, Culex, Aedes, and Mansonia species. So we have seen this in the last week assignment problem about the lymphatic filariasis. So we have seen this story that how this particular mosquito is spreading infection from non-infected person to an healthy, spreading infection from an infected to an healthy individual. So this mosquito have completes its life cycle. This particular worm, that is the filarial worm, completes its life cycle in mosquito as well as human. So it have to host for completion of the life cycle and for the spread of the disease. So this we have seen in the last week. If you want to study this in detail, you can check it out the video for last week. Then after that, we have seen some questions related to mucosal surfaces, that is the mouth and respiratory tract, gastrointestinal tract, female urogenital tract, and the male urogenital tract. We have seen some questions related to this mucosal surfaces, and we have seen this particular tissue, that is the MALT, that is known as mucosal associated lymphoid tissue. And we have also seen this, how this is in causing, how this is preventing us from the disease, from the transfer, like from the entry, how it is blocking the entry of foreign pathogen into our body. So we have also seen the secretory mechanism and epithelial barrier function of this mucosal surfaces. Then we have seen this transfer experiment in which we have seen that how a group of transgenic mice was used to study that particular cell type plays a crucial role in defense against virus infection. So we have seen this experiment also, and why this irradiated mice are used in immunological research that we have seen, like they are used for bone marrow transplantation studies and adaptive immunotherapy research and xenotransplantation studies and other infection and vaccine related research. After that, we have seen in the last week some questions were related to M cells in the immune cells. So we have seen the role of M cells, that is the transcytosis of antigen and the initiation of mucosal immune response. Then we have seen the role of Langerhans cells in the skin and its functions, that is the phagocytosis of pathogen and antigen processing and presentation via MHC class 2 molecules. Then we have seen immune surveillance mechanism also. Then we have seen this different types of cells, that is the totipotent cell, pluripotent, unipotent, and multipotent. We have seen difference between all these type of cells and what type of tissue, organism, or an individual can be generated from this particular cells. So we have seen a difference between these cells. So these were the few of the topics that we have covered while solving the last week's assignment. And today we will be solving the week 3 assignment. The due date is already over on 12th. So I hope you all have submitted this assignment. Can you please confirm whether you have submitted this assignment? Have you submitted this assignment, Shubhalakshmi? Am I audible? Okay, I hope you might have already submitted because the due date is over. And if not, then also you can see the solution to all those questions. So, in the week 3 assignment, the first question was like, what is the primary role of neutrophils in acute inflammation? So, anybody would like to answer? Like, what is the primary role of neutrophils in acute inflammation? Okay, so the answer for this question was option B, to swiftly respond to infections and clear pathogens effectively. Here we want to know what is the primary role of neutrophils in the case of acute inflammation. So as we know that neutrophils are the first responders in acute inflammation and they rapidly migrate to the site of infection or injury where they perform phagocytosis. That is, phagocytosis is the process of engulfing and destroying pathogens. And these neutrophils also release enzymes, reactive oxygen species, and other antimicrobial peptides to eliminate the microbial threat in order to protect the humans. So, these neutrophils contribute to the inflammatory response by releasing cytokines that recruit other immune cells. So, the correct answer is to swiftly respond to infections and clear pathogens effectively. And if we see other options, like option A was to produce antibodies against pathogen. So, this is not the function of neutrophils. Antibodies are produced by B cells, not by the neutrophils. And the option C was to facilitate blood clotting at the site of injury. So, you know how clotting is occurring at the site of injury. Platelets play an important role in the clotting. and preventing the blood flow. So this is also not a function of neutrophils. The next option was to promote tissue repair following injury. So as we have already seen in the last class, we had a discussion regarding macrophages. Two type of macrophages were there, M1 and M2. So there we have seen that macrophages play a larger role in tissue repair after the injury. So this is also not the function of neutrophil. And then the next option was to activate other immune cells in the lymph node. So this is also not the function of neutrophil. Which cells play a role in activating other immune cells in the lymph node? One is there. So which cells plays a role in activation of other immune cells in the lymph node? This is a function of dendritic cells and T cells, which are involved in the process of activation of other immune cells in the lymph node. So it is also not a function of neutrophil. So the correct answer for this question, like what is the primary role of neutrophils in acute inflammation, is to swiftly respond to infections and clear pathogens effectively. So correct option is B. I hope this question is clear to everyone. If not, then you can write in the chat box. We'll discuss it again. So see, there are different types of blood cells like monocytes, lymphocytes, neutrophil, eosinophil, basophil. These are all types of WBC. So other than that, there are macrophages and erythrocytes and platelets. And there are granulocytes, which are a category of WBC. These cells have granular cytoplasm. So types of granulocytes are neutrophils, basophil, and eosinophil. Out of which neutrophil and eosinophil can phagocytose the pathogen. So if the question comes, which cells can phagocytose a pathogen? So the answer will be neutrophil and eosinophil. So neutrophils are like 14, around 11 to 14 micrometer in diameter with multi-lobe nucleus with granules and cytoplasm. And it constitutes 50 to 70% of total circulating WBC and remains for 7 to 8 hours in blood and then migrates to tissue, and its lifespan is for about 3 to 4 days, and it is also known as polymorphonuclear leukocytes, like PMN is also called. And neutrophil is stained by both acidic and basic dye. See, as we can see in the structure, it have a multi-lobe nucleus, and there are some like molecules are there like glycogen, and some secondary granules are there, some primary azurophilic granules are there, and there are some phagosomes inside that. So this is a typical picture of neutrophils, and its immunological function of neutrophil is phagocytic role in acute inflammatory response, and it is the first immune cells to respond in inflammation. So this was only the question that we have seen here. Like what is the primary role of neutrophil in acute inflammation? So it is to swiftly respond to infection and clear pathogens effectively. So the main function of neutrophil is to phagocytosis in the case of acute inflammatory response, and it is the first immune cells to respond in case of inflammation. So this is the function of neutrophils, and I hope you might have seen the video lecture where the detailed function of neutrophil is mentioned. These neutrophils are also called PMNs. PMNs are nothing but polymorphonuclear cells. So these are the most abundant type of WBC and are prominent cells in pus and accounts for its whitish appearance. and it responds quickly following tissue injury, and it is a hallmark of acute inflammation. And these it has granulated cytoplasm stained with both acidic, that is the eosin negatively charged dye, and also the basic dye, that is the methylene blue, that is positively charged. And the granules are antibiotic in nature and contain defensin, saprocidin, cathelicidin, bacterial permeability inducing proteins that are BPI, hydrolases, and lysozymes. So these are like 50 to 70% of WBC, and these are multi-lobe nucleus. These have multi-lobe nucleus. Like see here, we can see in the structure the nucleus is multi-lobed. That is the multiple small compartments are there in the nucleus. So it is called as multi-lobe nucleus. And these are professional, highly phagocytic in nature, and these have short lifespan of like around 2 to 3 days, and these express these PRR like pattern recognition receptors. These cells also produce reactive oxygen species, also known as ROS, and the leukocytosis and dipediasis. That is the increase in neutrophil in circulation upon infection. These conditions, like question may come like what in what condition the increase in neutrophils in circulation upon infection, that condition is known as leukocytosis and dipedesis. So the next question is, what role does genetic similarity play in syngenic mouse development? So anybody would like to answer this question? Like what they are asking what role does genetic similarity play in syngenic mouse models? So here the second question is, what role does genetic similarity play in syngenic mouse models? So answer to this question is, it allows successful cell transplantation without the risk of immune rejection, aiding research on immune cell functions. So let's see what is this. Like what are syngenic mouse? What are syngenic mouse models? Syngenic mouse models are genetically identical or nearly identical, usually derived from inbred strains. And because of their genetic similarity, cells, tissues, or tumors can be transplanted between individuals of the same strain without being rejected by the immune system. And this makes them particularly valuable for research in areas such as cancer immunology, transplantation, and other immune cell function studies. Like we have seen in last class about the transfer experiment, where they have taken a group of syngenic mice, then they have infected particular small group of mice with the virus, and they isolated the discovered cells, like some of the immune cells or the produced immune cells. Then in other case also they have taken small groups of these particular syngenic mice. So these mice are mainly used in the immunological studies, like for the transplantation biology or for like transfer experiments or for studying the immune cell function experiments. So what happens here in the syngenic model is like they are taking some this cancer cells, that is the inbred cancer cells, then they are injecting these cells into immunocompetent mice, And after a time of 4 weeks, a syngenic tumor is developed in this mice. So if they are injecting these cells into a group of mice, then we will be having a group of syngenic tumor containing mice, like the syngenic mice. So what happens? Like what are exactly these? These are the mouse models genetically identical or nearly identical, usually derived from inbred strains. So there are some other models also, like genetically engineered model, carcinogen induced model, and some patient derived xenograft models. There are these also, but here they are asking about syngenic mice. So the question was, what role does genetic similarity play in syngenic mouse models? So the correct answer is it allows successful cell transplantation without the risk of immune rejection, aiding research on immune cell functions. If we see other options, like option A is it increases their lifespan, making them ideal for long-term studies. So there is nothing related to the lifespan. Like genetic similarity does not extend any lifespan. So this option is wrong. And the second option is it enhances their immune response, providing a model for robust immunity. So what about this option? Like it enhances their immune response. No, there is nothing like that. Their immune response is not inherently stronger. This is also wrong option. Then the next option is it reduces their sensitivity to pathogens, making them ideal for infection studies. This is also wrong option. There is nothing like that. The next option is it induces a rapid production of antibodies. There is nothing to do with the production of antibodies and syngenic mouse. So this is also wrong option. So the correct answer for this is it allows successful cell transplantation without the risk of immune rejection, aiding research on immune cell functions. The next question is what is the primary advantage of creating single knockout mice before double knockouts? So what would be the answer for this? Like what is the primary advantage of creating single knockout mice before double knockouts? What they are asking here, what is the primary advantage of creating single knockout mice before double knockouts? Knockouts are like removing something from that particular mice. So the correct answer to this question is it simplifies the breeding strategy for achieving double knockouts, making genetic studies more efficient. So let's see how this happens, what it want to say. Creating single knockout mice first makes it easier to breed and generate double knockout mice. So, by first establishing stable single knockouts, we can crossbreed heterozygous or homozygous single knockout mice to efficiently produce double knockout offsprings. So, this approach ensures a controlled and systematic breeding strategy, reducing complications that might arise if both genes were targeted simultaneously. And this method also helps to confirm the viability. Some double knockout might be embryonically lethal, so understanding single knockout effect is useful. Like directly, we should not do the double knockout because that might be the lethal for some these models. So first we need to study the effect of single knockout. Then we can go for double knockout. So for confirming the viability, it can be used. And for the optimization of breeding efficiency, like for establishing single knockouts, first streamlines the process of generating double knockout without excessive back crossing. Like we can optimize the breeding efficiency by using a single knockout by removing one gene. Then we can, if it is successful, then we can go for the double knockout. So it is optimization procedure. And for improving the reproducibility, like single knockout line serves as stable genetic resource for further experiments. Like we can check it out whether how single knockout works. Then we can go for double knockout. That is for improving the reproducibility. So the next question is, what is the significance of the density gradient centrifugation method in immunological research? What is the significance of density gradient centrifugation method in immunological research? So the correct answer is it isolates peripheral blood mononuclear cells, that is PBMCs, by providing a reliable source of immune cells for experimentation. Like what is a density gradient centrifugation? What happens in this? Like density gradient centrifugation is a technique for immunological research for the separation and isolation of PBMCs. PBMCs are peripheral blood mononuclear cells. So we can isolate these PBMCs from whole blood. And what are these PBMCs? These PBMCs include lymphocytes, that is T cells, B cells, NK cells, and monocytes. So these are the PBMCs. So we can isolate these cells for our immunological research. So what happens in this particular centrifugation technique? What we actually do is like we use a density gradient medium. Generally, we call bake or like glycerol density gradient, like for glycerol or ficol we can use, or percol we can use. These are some density gradient chemicals. And then we centrifuge to separate the blood components. So the cells will be separated on the basis of their densities. Like see here, we can see that these PBMCs containing lymphocytes are from 70 to 90%, then monocytes 10 to 30%, and some of the dendritic cells are also there. So this density gradient centrifugation separates by using histopag 1077, which contains polysucrose and sodium ditrioxide. So what we do, we take this, we call histopag, that is the density gradient medium. We put our blood sample onto this. Very slowly we add this. Then we perform the centrifugation. After centrifugation, what we observe that the RBCs, that is the red blood cells, those are the highest density cells. These form a layer at the bottom. Then this medium, the density gradient medium, forms an intermediate layer that is the ficol. And just above this density medium, there is a buffy coat containing PBMCs. A very thin layer will be there containing the PBMC cells. And above this PBMCs, there is a layer of plasma. So this plasma will form the topmost layer, followed by these PBMCs will form a very thin layer here at the intersection. Then the density gradient medium will be there, and finally the RBCs will form a palette at the bottom because those are the highest density cells in blood. So this is the density gradient centrifugation. So what happens here, we can see clearly from this beautiful diagram here. Like the density of cells and components in this portion is like in this upper portion is less than 1.077 gram per ml. Why this 1.077 gram per ml? Because this is the density of this density gradient medium. That is the Ficol or whatever we are using. The solution density is 1.077 gram per ml. So the cells who have the density less than this will form a layer above this. That is the upper layer. This upper layer will contain, it is a plasma layer that is containing the 55% cells, 55% covering the 55% portion of this tube, which contain thrombocytes, that is the platelets, cytokines, hormones, and electrolytes. Then just below this, there will be a very thin layer at this intersection that will be of PBMCs, that is the mononuclear cells. These mononuclear cells contain this lymphocyte and monocytes. These will be here in this thin layer. Just below this density gradient medium will form a layer, and just below the density gradient medium, there will be like cells which have the density more than 1.077 gram per ml. Why? Because these are heavier than the density gradient medium, so those will settle down at the bottom. So these will contain like these erythrocytes, like majorly RBCs and this some of the polymorphonuclear cells, which are the eosinophils and neutrophils, because these are the higher density cells. So it will be at the bottom. So what was our question? Question was, what is the significance of the density gradient centrifugation method in immunological research? So it is used to isolate PBMCs, providing a reliable source of immune cells for experimentation. So B is the correct answer. If we check other options, so first option is it is used to transport blood samples. No, this is not a transportation technique. It is a separation and isolation technique of blood cells. So this first option is wrong. This option is it enhances the growth of RBCs. No, it does not promote any growth of RBC. It is a method for measuring blood viscosity. No, blood viscosity is measured by some other techniques, not by this density gradients. It is for separation on the basis of density. Separation of blood cells on the basis of density. Next option is it allows for the direct observation of immune cell functions. No, this method isolates cells but does not directly assess their function. Like we can only separate and isolate the blood cells. We cannot say anything about their immune function. So this option is also wrong. So the correct answer is it isolates PBMCs and provides a reliable source of immune cells for experimentation. I hope this answer is clear to all of you. If not, then please let me know. Okay, let's move to next question. The next question is which of the following statement about NK cells are true? Like, what is this next question asking? It is the which of the following statements about NK cells are true? The options are see the correct options. Take a time, take 2 3 minutes and explore all these options. What is true about NK cells, that is the natural killer cells? There are so many options for this. So the first correct option is NK cells induce apoptosis in infected or cancerous cells using perforin and granzymes. Yes, it is absolutely correct answer because these NK cells release perforin, which creates pore in the target cell membrane, and the granzymes, which enter and trigger the process of apoptosis for cell killing. So this is the correct answer. Like NK cells have perforin and granzymes. And the next correct answer is the activation and inhibition balance of their receptors is crucial for targeting abnormal cells. So what this option is saying, like these NK cells have maintained a balance of activating and inhibitory receptors to determine whether to kill a target cell. So if inhibitory signals are absent or weak, so then NK cells will attack. So the activation and inhibition balance of the receptors is very crucial for targeting the abnormal cells. Like the balance of these type of receptors will decide whether to kill the target cell or what to do next. Next option is NK cells can be activated therapeutically for cancer treatment. So yes, this is correct answer. The NK cells are being explored currently in immunotherapy, including cytokine-based activation or checkpoint inhibition and NK cells-based some transfer therapies. So NK cells are used actively for this therapeutic. Measuring lactate dehydrogenase release can access NK cell activity. Yes, when the NK cells lies the target cells, like when they kill the target cell, this lactate dehydrogenase enzyme is released, which is commonly measured in the cytotoxicity assays also. So by measuring this lactate dehydrogenase enzyme release, we can access that NK cells are performing their activity. So let's also discuss these incorrect options, like why they are incorrect. The option B was they primarily respond to peptide antigen similarly to conventional T cells. No, unlike T cells, NK cells do not recognize specific peptide antigens via T cell receptors, but they detect missing or altered self-MHC molecules. So they will not respond to this peptide antigens like T cells. The next incorrect option is option D, that is NKT cells do not play a role in bridging innate and adaptive immunity. This is wrong because they play a role in bridging the innate and adaptive immunity. They do not utilize cytokines in their immune response. No, this is also wrong. These NK cells utilize cytokines Like you might be studying in the next unit, that is the cytokines and other immune mediators. In that unit, you will study that these NK cells utilize cytokines in their immune response. Like they utilize this some of the interleukins and interferons for the activation and functioning of these NK cells. The next option is they exclusively act without any assistance from other immune cells. No, NK cells will not do alone any job. They will interact with the dendritic cells, macrophages, and T cells via cytokines and signaling pathways. Then they will do the immune regulation. So this is also wrong option. So these are the correct options. Like what are what is true about NK cells? So this NK cells induce apoptosis in infected or cancerous cells using perforin and granzyme. And the activation and inhibition balance of the receptors is crucial for targeting abnormal cells. NK cells can be activated therapeutically for cancer treatment, and measuring the level of lactate dehydrogenase release can assess NK cell activity. Next question is dendritic cells are primarily derived from and dendritic cells are derived from which lineage? So it is very basic and very direct question. So the answer to this is from both the lymphoid and the myeloid lineage. And we have seen in this lecture video also that this hematopoietic stem cell is forming two type of lineages. One is this myeloid progenitor cells, other one is lymphoid progenitor. And from this myeloid progenitor, dendritic cells are developed, and from this lymphoid progenitor also the dendritic cells are developed. So dendritic cells are primarily derived from both the lymphoid and the myeloid lineage. So you might have seen this beautiful chart of which cells are formed from which lineage and what progenitor cell is forming which cells and what are the categories of granulocytes and granulocytes. And you might have seen this in the video. If not, then please go through the video lecture of this particular session, the cells of immune system. The next question is, which of the following statements about macrophages are true? Like, which of the following statement about macrophages? Like we have seen about dendritic cells, NK cells, and now this is about macrophages. In last class also we have discussed so many things about macrophages. So please recall those points what we have discussed and then see the options which are given here. See, these are the correct answers, correct options for about the macrophages. So the question is, which of the following statements about macrophages are true? So the correct options are macrophages are derived from myeloid lineage. Yes, we have just seen that this macrophages here are derived from this myeloid progenitor cells or the myeloid lineage. Macrophages are derived. So first, this is the correct option. Then this macrophages can be activated by LPS. What is LPS? LPS is the lipopolysaccharide, which is present in the gram-negative bacterial cell wall. So what they are saying here that macrophages can be activated by LPS. So yes, it is correct option. So like LPS, like lipopolysaccharide from the gram-negative bacteria, binds to this toll-like receptors and they trigger the cytokine release. So this macrophages can be activated by LPS. Next option is macrophages play a role in maintaining homeostasis by scavenging dead or damaged cells. Yes, macrophages remove the apoptotic cells and debris via the process of phagocytosis and prevent the unnecessary inflammation and maintain the tissue function. So yes, this is also correct option that macrophages play a role in maintaining homeostasis by scavenging dead or damaged cells. Next one is macrophages produce reactive oxygen and nitrogen species to kill pathogen. Yes, this is also correct option. Macrophages just produce the reactive oxygen species like hydrogen peroxide, superoxide, and also some of the reactive this nitrogen species like nitric oxide, and which are toxic to bacteria and other pathogens. So macrophages do produce these cells for the killing of the pathogen. So these are the correct options for what is true about this macrophages. The correct options are macrophages are derived from myeloid lineage. Macrophages can be activated by LPS. Macrophages play a role in maintaining homeostasis by scavenging dead or damaged cells. Macrophages produce reactive oxygen and nitrogen species to kill pathogens. Let us also see the incorrect options. The option A is macrophages are involved in tissue repair. We have discussed in last class about the tissue repair and phagocytosis role of macrophages. There we have seen that these macrophages are of two types, M1 macrophage and M2 macrophage. So not all the macrophages are involved in the tissue repair. Like if it was like some macrophages are involved in tissue repair, then it might be correct option. But here it is written only that the macrophages are involved in tissue repair. So we cannot say that this is correct because only the N2 type of macrophages, those promote tissue repair, whereas the N1 type of macrophages are pro-inflammatory and they destroy the pathogen. So this is the half statement, so it is incorrect. And the next statement is macrophages cannot phagocytose pathogen or cellular debris. This is the wrong statement as it is well known, popularly known that macrophages are the professional phagocytes that engulf pathogen and cellular debris. So this is wrong. Next option is macrophages primarily reside in the blood and do not differentiate in tissues. So this is wrong, like because macrophages circulate in the blood. They are not primarily reside in the blood. And these can differentiate in tissues into different cells. Like if you might have seen the lecture video of macrophages and gone through the study material, then you will find there that these macrophages are different in different tissues. Like they differentiate in tissues. Like these macrophages in the liver are called the Kupffer cells. In the brain, those are known as the microglia cells. And in the lungs, those are called as alveolar macrophages. So they do differentiate in tissues. So this is also wrong statement. The next statement is macrophages are involved in B cell development. No, this is wrong because macrophages can influence the adaptive immunity, but they do not directly contribute to the B cell development because this B cell development happens in the bone marrow. So macrophages are not directly involved in B cell development. So this is also wrong answer. So the correct statement for this macrophages are derived from the myeloid lineage. Macrophages can be activated by LPS. Macrophages play a role in maintaining homeostasis by scavenging dead or damaged cells. And macrophages produce reactive oxygen and nitrogen species to kill the pathogens. So these are the correct options. Next, very small and very direct question is which marker is associated with cytotoxic cells? Which of the cell surface marker is associated with the cytotoxic cells? It is very direct. Cytotoxic T cells contain CD8 on its surface. And this cytotoxic T cells are they do possess the CD8 receptor on its cell, which binds to the MHC molecules for antigen presenting and processing. So the correct option is CD8. Next option is which receptor is primarily involved in the activation of mast cells, basophil, during allergy reaction. So like this is which receptor is primarily involved in the activation of mast cells and basophil during allergy reaction. See the correct answer for this question is Fc epsilon R1. That is Fc epsilon receptor 1. R1 class is primarily involved in the activation of mast cell and basophil during allergy reaction. See, we have just discussed about the dendritic cells, NK cells, macrophages, and now this question is about mast cell and basophil. So this Fc epsilon R1 is a high affinity receptor for IgE, that is the for antibody for the allergy, that is IgE, which is primarily expressed on mast cell and basophil. So this Fc epsilon R1 is expressed on mast cell and basophil. This receptor plays a role in the allergy reaction and hypersensitivity response, that is in you will see in more detail in the later units. But you might have seen or studied about this allergy reaction with respect to the mast cell and basophil. So this IgE binds to the Fc epsilon R1 on mast cells and basophil after being sensitized by the allergens. So the subsequent allergen exposure leads to the cross-linking of this IgE molecule, which triggers the intracellular signaling. This ultimately results in the degranulation of mast cell and basophil, which release the histamines and prostaglandins and like other molecules which are present in mast cell and basophil in response to the allergic reaction. And these mediators cause the vasodilation, increased vascular permeability, and smooth muscle contraction, which leads to the allergy symptoms like anaphylaxis, asthma, and some other conditions. So, this is the brief mechanism of how IgE mediates the allergic reaction via mast cells and basophils, as these cells contain the like granular molecules like histamine and prostaglandins. So, upon interaction with the IgE, these will degranulate and release its content, which these mediators will cause further downstream reactions and stop the allergy. And what about other options like this Fc gamma R3, Fc alpha R1, TLR4, CD16? See, this Fc gamma R3 receptor is a receptor for IgG, that is the immunoglobulin G, primarily involved in NK cell mediated ADCC, like antibody dependent cellular cytotoxicity. Like this Fc gamma R3 will come into play when we'll talk about the receptor for IgG involved in NK cell mediated response, not in the this allergic reaction. In allergic reaction mediated by IgE with interaction to mast cell and basophil, the receptor will be Fc epsilon R1. When we talk about this NK cell mediated activity with respect to the IgG antibody, then this Fc gamma R3 will come into play. And when we talk about this Fc alpha R1, so this is a receptor for IgA, immunoglobulin A, which is found on neutrophils and monocytes, and it plays a role in mucosal immunity. Like we have seen in our last class about the mucosal immunity that is mediated by IgA, that is immunoglobulin A. So at that place, this Fc alpha R1 receptor will come. And this TLR4, it is involved in innate immune response against gram-negative bacteria. This is toll-like receptor TLR, and it is not to do anything with allergic reaction. And the CD16, it is also known as the Fc gamma R3. Like this Fc gamma R3 is involved with the NK cell activation via the IgG antibody. It is not involved in mast cell or basophil. So when it comes about mast cell and basophil, then the correct option will be Fc epsilon R1 receptor. Last question for week 3 assignment was, which enzyme is responsible for the production of reactive oxygen species in neutrophils? Which enzyme is responsible for the production of reactive oxygen species in neutrophils? We have seen it in the previous slides, this NADPH oxidase. Like in initial slides, we have seen that this neutrophils can form reactive oxygen species or the nitrogen species. So here this NADPH oxidase is the primary enzyme responsible for generating reactive oxygen species in neutrophils. During the oxidative burst, that is the respiratory burst you might have heard about. So this enzyme complex facilitates the conversion of oxygen into superoxide anions, which is subsequently leads to the production of other reactive oxygen species such as hydrogen peroxide. And these ROS are very important for killing pathogens inside the phagolysosome. So this NADPH oxidase is the main mediator enzyme for this reaction in neutrophils. So there is a procedure for the generation of this reactive oxygen species. I will briefly explain that. For the activation of like this NADPH oxidase enzyme is activated upon neutrophil stimulation, and the superoxide is produced from the molecular oxygen, and this will ultimately lead to the production of hydrogen peroxide, which will kill the pathogens. So this is about this NADPH oxidase enzyme. And if we see other enzymes like this superoxide dismutase, so this enzyme, the superoxide dismutase, it converts the superoxide into hydrogen peroxide but it does not directly generate the reactive oxygen species. It is for the conversion of superoxide into hydrogen peroxide, but it will not produce the reactive oxygen species. And this catalase enzyme, this breaks down the hydrogen peroxide into water and oxygen. So it is used for the reduction of reactive oxygen species level, like for reducing the level of ROS, catalase is used. It works in just opposite way to NADPH oxidase. Like this oxidase is responsible for production of ROS, and this catalase is responsible for reducing ROS level. Next is glutathione peroxidase. So this glutathione peroxidase, it detoxifies the ROS and prevent the oxidative damage to cells. Like this do not reduce any ROS, but it detoxifies ROS, like it removes the ROS and prevent the oxidative damage to the cells. And the next enzyme, last enzyme is lysozyme. So it is an enzyme for degrading bacterial peptidoglycan. It have nothing to do with the ROS production. So if the question is about which enzyme is responsible for the production of ROS in neutrophils, then the correct option will be NADPH oxidase. If it's about the removal of ROS or for reducing the level of ROS, then it will be catalase. And so these are different type of enzymes. So do not get confused with this enzymes. So these were the questions for week 3 assignment. I hope these are clear to you. If not, then you can write in the chat box or you can post a comment below the video or you can post your query directly at the discussion forum of the NPTEL. So we can respond to your query there or you can comment down the video lecture so I can take it out from there also. I'm happy to explain any of the question again. If it is not clear to anyone, please feel free to ask. So this brief summary of what we have seen in week 3 assignment. We have seen these questions, the role of neutrophils in acute inflammation we have seen about different type of blood cells. We have seen a detailed explanation of the neutrophil morphology and its function. Yes, we have seen summary of neutrophils. Then we have discussed about syngenic mouse model, why they are used and what are the syngenic mouse model, where we can use. We have seen this with an example of this transfer experiment, which is already discussed in detail in the last week video. And we have seen about other models like syngenic model, genetically engineered model, carcinogen-induced model, patient-derived xenograph model. Then we have seen about single knockout and double knockout, what are their advantages, why they are used. Then we have seen the details of this density gradient centrifugation method, which is used for the isolation of PBMCs, which are peripheral blood mononuclear cells. And we have seen what type of this density gradient medium are used and what is the principle behind this, what is the density of this density gradient medium, and what are the layers formed in a whole blood sample, and each layer contain which type of cells that we have seen. Then we have seen the detailed function of the NK cells, which are natural killer cells. And then we have discussed the dendritic cells, the primary role of these dendritic cells, from where they are originated, from which lineage. Then we have seen how hematopoietic stem cells form two different lineages, which ultimately forms multiple type of cells. Then we have seen the function of macrophages. There are different activity and function of macrophages. And we have seen this cytotoxic T cells, which receptors are present on the cytotoxic which is CD8. Then we have seen this mast cells and basophils, how these are involved in the allergic reactions, and which surface receptor are present on this mast cell and basophils. And we have also seen where these other type of receptors are present, like this Fc gamma R3 receptor, Fc alpha R1 receptor, TLR CD16, where these receptors are present and what are their functions that we have seen in detail. After that, we have discussed the production of reactive oxygen species in neutrophils, like which enzyme is responsible. And parallelly, we have also seen about the other enzymes, where these enzymes are produced and what is their function. So these were the week 3 assignment questions. I hope all the questions are clear to everyone. If not, please write in the chat box. So, okay, if no doubt, then we can wrap it up the session for today. You can check it out this video on the YouTube, and also the NPTEL link will be shared to you. In next class, we will be discussing the week 4 assignment questions. If you have any doubt, then you can post comment in the discussion forum. Okay, thank you everyone. This is all for today. Thank you.